Hello everyone, welcome back to my video tutorials for Pesudo-bulk RNA data analysis. In video 1, we prepared a thread object from 6 human non-single RNA sequencing dataset, and we subset the fibroblast cluster and saved the individual thread object. So today, I'm going to use this thread object and show you how to use classic edgeR pipeline to do Pesudo-bulk RNA sequencing data analysis. So first, let's load the thread object. You can see we have a thread object here named the Fibra. We can have a look at the metadata for this thread object. You can see the row names are the barcode. We have the sample information in the origin ident, then uncount and then feature RNA. So here the cell cluster information. Because we subset the fibroblast cell cluster, so all the cells in this thread object will be fibroblast. So the sample information is more important for today's demonstration. We have 3 controls and 3 IPF dataset. So let's use the dim plot to have a look at the sample types. You can see we have 3 control 1, 2, and 3, and 3 IPF 1, 2, and 3 dataset in this fibroblast thread object. And we can use the table function to see how many cells in each dataset. Let's run. You can see the cell numbers in each dataset. The dataset control 3 has the smallest number of cells, only 43 cells. Later, when we perform the dataset clustering, you will see this dataset will show long distance from the other control dataset. So next, we can set the default assay as the RNA to create Pesudo-bulk samples. So now the thread object is ready for creating Pesudo-bulk samples. We are going to use the thread to Pesudo-bulk function from the edgeR package. If we run this function on the thread object, we will create Pesudo-bulk samples by aggregating read count together for all the cells with the same combination. Here we only have the fibroblast. So the thread to Pesudo-bulk function has 3 arguments. The first one is the name of the thread object. Here we are going to use the fibroblast thread object. Then the second argument is the sample. Because we are going to identify differentially expressed genes between IPF and control fibroblast. And we know the sample information in the origin ident column in the metadata, so we can set here for the sample as origin ident. So I'm using the fibroblast cluster as an example to show you how to run Pesudo-bulk RNA analysis. You can subset the other cell types such as immune cells, endothelial cells, epithelial cells to perform the similar analysis. So now we can run the thread to Pesudo-bulk function to create Pesudo-bulk samples, and we named it as Y. So let's run this function for the fibroblast thread object. You can see we created a large DGE list object. We can click this object and have a look. You can see the list as the count, samples, and the gene information. The count contains 20,680 genes, and we have 6 samples. The samples not save the group information, library size, normalization factors. At the moment, you can see the normalization factor is 1 for each dataset. Later, when we perform the dataset normalization, you will see the changes for the normalization factors. We also have the sample information and the cluster information. Then down here is the gene slot. We have the gene information and the object we set as RNA. So now you know the data structure for the large DGE list. And you can use the gene function to look at how many genes and how many samples are there in this dataset. We can run. I already show you we have 20,680 genes and we have 6 samples. If you want to see the counts for the Pesudo-bulk RNA counts, you can get the counts from the DGE list. Let's get it. You can see the count is a large matrix data. We can click the count and have a look at the gene and the dataset information. You can see the gene names are the row names, and also we have 6 samples. It's the column name, control 1, 2, and 3, and the IPF 1, 2, and 3. Down here are the counts for each gene in each data set. So here is the Pesudo-bulk RNA count, and the data structure looks the same as bulk RNA sequencing dataset now. Next, we can have a look at the sample information in the DGE list. We can get it as samples. Then we can click the samples and have a look at the sample information. You can see we have 6 samples. Control 1, 2, 3, IPF 1, 2, 3. And all the cells in each dataset are fibroblast. And the first column is the group column. At the moment, it labeled as 1 for each dataset. Later, we are going to rename it as the control and IPF. The second column is the library size and then the normalization factors. In the sample column, we have the sample information and the cluster we know. They are fibroblast cluster for each dataset. So now we can change the group information. We can set the group as the control and the IPF. Then we can rename the groups in the DGE list dataset. And we can get the samples information again. Now, if we click the samples, you can see we change the group information to control, IPF, and IPF. So now we have correct group information. So we successfully created Pesudo-bulk RNA data for the fibroblast threat object. So in next step, we are going to filter the data and also normalize the data. So normally for edgeR analysis, we filter the library size below the threshold of 50,000. We can use the summary function to have a look at the library size in each dataset. You can see the minimum library size in this dataset is bigger than 50,000. So we don't need to filter the library size, but I'm going to run through the code for demonstration. In case the samples in your dataset has library size smaller than 50,000. So let's just run through the code here for demonstration. We can set the library size bigger than 50,000 as keep samples. Then we can have a look at the keep samples. You can see here, because the library size are bigger than 50,000 for all the samples, so all the samples are true. If we use the table to see the keep samples, you can see all 6 samples are true. Then we filter the samples for this dataset. Because all the library sizes are bigger than 50,000, even we ran through the code, we didn't filter out any samples. So next we can filter out no expressed genes. We are going to use the filter by expression function from the edgeR package. So the group will be the 6 samples. We know we have 3 control dataset and 3 IPF dataset. So we are going to filter out the genes that has minimum count below 10 and the minimum total count below 20. So let's identify the genes that have the minimum count below 10 and the minimum total count below 20. So now we can use the table function again to see the logic value for the heap genes. You can see we have 9,208 genes labeled as false and 11,472 genes labeled as true. So now we can filter the genes, just the keep genes that have high expression. So here we have 11,472 genes. So let's filter the data. We can run. So after the data filtering, we can run data normalization. So in edgeR for data normalization, we use the TMM normalization method to estimate the effective library size. TMM stands for trimmed mean of M values. So we can use the normalized library size function from the edgeR to normalize the data. We can run the normalization. After the normalization, we can have a look at the normalization factors now. You can see now the normalization factors will change from 1 to 0.9 for the minimum library size, and the maximum library size is 1.1576. So the small library size uses smaller normalization factor, and the large library size uses large normalization factors. So we filter the library size and the only express the genes, and also we normalize the library size. So next we can have a look at the data set. We can use the multi-dimensional scanning plot method to explore the data file for the Pesudo-bulk RNA data. First, we can get the sample information from the sample column in the DGE list. Let's run. Now we can plot the samples using the multi-dimensional scanning plot. We have 6 samples here. Let's run it again. You can see in the plot window, we have a MDS plot to show 6 samples. So let's add the sample information in the plot. And we can zoom in. You can see black, red, and green are control samples. Here are the black, red, and green. Then dark blue, light blue, and purple are IPF samples. So 3 IPF samples are here. You can see 3 IPF datasets were clustered on the right-hand side, and 3 control datasets were clustered on the left-hand side. I showed you before the sample 3 from control samples has very small cell number. It only has 43 cells. Then the sample 3 has long distance from sample 1 and sample 2. So from the MDS plot, you can clearly see we can separate the IPF samples from the control samples by plotting the Pesudo-bulk RNA data. So the distance between IPF samples and the control samples indicate our Pesudo-bulk RNA analysis is working. So next, we need to estimate the dispersion. Because in today's video tutorial, we are going to perform pairwise comparison using classic edgeR pipeline. So we don't need to design a matrix for today's analysis because we perform pairwise comparison. for IPF against the control Pesudo-bulk RNA data. So when we run the estimated dispersion function, we just need the name of the DG list. We don't put any other arguments here. In my next video, when I show you the complex edgeR pipeline, you will see we need the design matrix argument in here. So before we run the estimate dispersion function, you can see we only have 3 elements in the DG list. Now we can run the estimate dispersion function. So it is done. You can see edgeR using the classic model. And now we have 11 elements in the DG list. We can have a look at the DG list now. You can see when we run the estimate dispersion function, we obtain the common, trended, and tangle-wise dispersion value for each dataset. You can see the value here. If you want, you can also get the value. For example, we can get the common dispersion value from the DG list. You can see the common dispersion value is 0.13. Then we can plot the biological coefficient variance to see the BCV information for all the genes. We can run. You can see we generated a plot in the plot window. It shows the tangle-wise, common, and trend dispersion value. The y-axis is the biological coefficient variation, and the x-axis is the average log count per million for all the genes. So now the data is ready to test for differentially expressed genes. Because we only compare the IPF and the control fibroblast, and we are using the classic edgeR pipeline, so we can use the exact test to identify the differentially expressed genes between IPF and the control. So the exact test is based on the contour-adjusted condition maximum likelihood method. Because we only have 2 conditions. In the group, we have the control and IPF. Then we can set the pair argument as control and IPF. If we set in this order, we will identify the differentially expressed genes in IPF against the control samples. So let's run the exact test. You can see we generated a large DGE exact test object. And again, it contains 3 elements. We can click and have a look. You can see the 3 elements. The first one is the table. The second one is the comparison. We compared the IPF sample with the control sample. And the third one is the gene information. The gene information is exactly the same as the DGE list. Then in the table data slot, you can see we have the log FC value for each gene, and the values log count per million for each gene, and also the p-values. So for identify differentially expressed genes, the information in the table is important. So we can generate a table to show the log FC, log CPM, and the p-value as the DGs. So let's run. You can see we have the DGs data frame here. We can click it and have a look at the data frame. You can see the row names are the genes. We have the first column as log FC, second column is log CPM, the third column is the p-value. At the moment, we have the log FC, log CPM, p-value for all the genes. We analyze the 11,472 genes. But not all the genes have significant changes between IPF and control samples. So we can just keep the genes that have p-value below 0.05. Then we can use the filter function to filter the genes. Let's run. You can see now in the new dataset we named as IPF-DGs, we only have 2045 genes that show significantly changes. Once again, we can click the dataset and have a look at the genes that show significant changes. We can click the log FC column to reward the field changes. Now you can see the upregulated genes in IPF dataset. You can see collagen 10A1, AL13RA2, and MMP11 pyroxene. We see those gene upregulation when we perform the single-cell RNA sequencing data analysis. Down here, we can also find the upregulation of collagens. You can see here collagen 1A1, collagen 11A1, and also the upregulation for the CTHRC1 gene. This is a marked gene for myofibroblast in human, also mouse fibrotic lung. Further down, you can see gene downregulation in IPF fibroblast compared to control cells. So from the upregulated and downregulated genes, we know our Pesudo-bulk RNA sequencing analysis generated a reliable result. So now we can use the feature plot to look at the upregulated genes, for example, pyroxene in the fibroblast cluster. We can run the feature plot. Let's zoom in. You can see the upregulation of pyroxene in all three IPF datasets. And we can use another sample, the CTHRC1, because it is a marked gene for myofibroblast. We can run the feature plot again for CTHRC1. Let's run. And we can zoom in again. You can see the upregulation of CTHRC1 in IPF dataset compared to control dataset. So now we can save the identified DGs as a CSV file, and I'm going to save it in my edgeR folder. Let's run. And we can go to my edgeR folder and have a look. You can see we saved the identified differentially expressed genes between IPF and control dataset. Then you can use this dataset to perform more analysis, such as Go and KEGG signaling pathways analysis, which I'm not going to demonstrate today. So I'm going to stop from here for today's demonstration. I hope this video tutorial can help your data analysis. As I mentioned, I only used the fibroblast cluster for the demonstration. You can run the analysis for your dataset and the cell types that you are interested in. So please subscribe my channel if you haven't to do so. And in my next video tutorial, I'm going to show you more complex edgeR pipeline. I hope to see you in my next video tutorial.